Breaking the wall of biocomplexity. How a reactive systems approach may lead to full dynamic models of multi-cellular organisms. David Harel, the Weizmann Institute of Science.
On November the 9th, I visited my grandmother in Leeds for a few hours. We heard the news together in her nursing home. She passed away peacefully a couple of weeks later. In a way, the falling of the wall was a symbolic circle being closed for her, since she arrived in the UK as a toddler with her parents after fleeing persecution in the Ukraine.
So I'm a computer scientist, but I'm going to be talking about biology and essentially about building computerized models of biological systems. And one of the questions we ask ourselves is, why do we even do computerized modeling? What should we model? How should we do the modeling? And one of the most interesting questions is, how do we know when we've built a model that the model is complete and is valid? These questions don't really have a lot of importance when you're building a human-made machine. If you're building a model of a telephone or of an aircraft, we know the answers to these questions easily, but they're especially acute if we want to build a computerized model of something in nature. If you want to build a model of the weather, for example, you know, how do we do it? Why should we do it? And the red question is very important. How do we know when we've finished and the model is valid? A key issue that underlies the kind of work that I do in this area is drawing a line between what we call a reactive system. In our field is a system whose complexity comes from things that you do. You press a button, the temperature rises above a certain level, a valve opens, something shuts down, you put your card into your bancomat, your ATM, it causes something to happen. This is a reactive system, and our claim is that what makes biology very complicated to understand are exactly the same kinds of things that make understanding an F-15 or Windows or some complicated human-made machine understandable. So here is a very complicated man-made or human-made reactive system, and here is a very complicated God-made or Buddha-made or whatever you want made, nature-made reactive system. And I like to say that the similarity between these two things is a lot greater than the difference. And if there is a major difference between an F-15, I think this is, and an elephant, the difference is that the elephant is orders and orders and orders of magnitude more complicated. But the difficulty in understanding one is very similar to the difficulty in understanding the other. And the whole idea behind the kind of work I'm talking about today is to try to use techniques that we use in order to build the things on the right in order to understand or reverse engineer the things on the left. So if you want a soundbite sentence, how about trying to reverse engineer an elephant or a worm or a heart rather than, say, engineering an F-15? Coming from the Middle East, I have other reasons to try to do less of the second here and more of the former. And some of the questions that arise are, what should we be modeling? The brief answer here is, my approach is, don't just take a question and try to answer it using a model, but try to do the whole thing. Be comprehensive. Which raises the two questions. I call them horizontal delineation and vertical delineation. Horizontal one is, what is the whole thing in terms of what is the borderline between what you're going to model and what is outside the model? And there are various possibilities that come up. One is try to do an entire cell. There was a wonderful paper published a few months ago about a model of a cell, but you know, some of my friends say to me, you're talking about elephants and worms, let's see you even do one cell or two cells. That's a good idea to try to do that. But then you can try to do a heart or a liver or a toenail or a tail or an organism, an elephant, for example, or a fly. But you can also think about doing a whole herd of elephants or you know, a colony of ants. The point right now is not to make a decision here, but to understand that you have to make it very clear what is going to be the system you're modeling and what is outside of it and is part of the environment. And then there is vertical delineation. You have to make a decision about the level of detail which your model is going to have to deal with. And you can say, I'm going to stop at the intercellular level. I'm going to talk about cells. I'm going to talk about communication between cells. Each cell will just be a black box. But then if you do that, it's quite obvious that you have to go to some extent inside the cells and talk about some of the major molecules they're in. And if you do that, people will say, well, you know, if you go there, you have to talk about genes and about DNA and so on. But I also have friends who say, if you go to that level, you really have to get into the biochemistry and maybe even into the physics, quantum effects, as we heard, string theory, whatever. And again, my point here is not to make a decision right now, but to explain to you that when you start out, you have to make decisions ahead of time about what level of detail you're going to have to deal with. You cannot talk about elephants and then someone will say, well, what happens if I turn off this gene? If you haven't told them ahead of time that you're not modeling on the level of genes. The crucial point, however, in doing comprehensive modeling of a complete biological system is this. You want to do elephants? Fine. Go ahead and interview all the world's elephant experts and put everything they know somehow consistently into your model. The interesting point is if you do that, you can do anything else that they don't know anywhere you want. So, for example, if you know the elephant is here and a month later it's here, this might be spatial movement, it might be developmental. No one seems to know how it goes from here to there. You're doing computerized modeling in silicon. You can, you know, buy some software and make it go like this. The reason that's okay is because if this gentleman comes along, he's our elephant expert, and he looks at the screen and says, no, it doesn't go like that, it goes like this. Then you have left out something that has been known about elephants from your model. If somehow you put consistently everything that's known into your model, you can make up the rest. Not that that's easy, but that's at least in principle possible in a computerized model. So the challenge here, the dream, the wall that I would like to break, or I would like someone to break, I call it the whole organism challenge, is to construct a full, correct, true to all known facts, four-dimensional model, not of a single cell, which is very difficult, and partly it's already been done, but of a multi-cellular organism, namely an animal. Full and correct are in quotes, because the full depends on the level of detail, the correct depends on something I haven't said yet. But in general, I hope you get the idea. We want it to be consistent with everything that's known. We want it to be seen in 3 dimensions, but to move and be animated over time, and so on. Some of the properties this model should have, I'll just run through them quickly. It should be realistic. It should show the elephant or whatever it is developing from a single cell and also moving around, eating, having breakfast, and so on. It should be interactively executable, modifiable, zoomable. Obviously, it should not just be a nice movie, but it should be mathematically precise and rigorous. But the mathematics should somehow be hidden under the surface so that biologists should be able to do the modeling. Someone once said to me, why don't you say even biologists? So that sounds kind of deprecating, but it's fine because biologists are trained to do things I have no idea how to do and vice versa. And yes, we want biologists to be able to do the modeling themselves, so a bunch of differential equations is probably not going to be the right kind of way to do the model, at least on the surface. Why? Why shouldn't we be satisfied with having a question that we want answered, building kind of a mathematical or computerized model around that, getting an answer and going on to the next thing? Why do we want to do, or why do I want to do the big thing? Well, scientific altruism. I'd like to truly understand life, and I think this is a wonderful way to understand life in detail, but there's a lot more to it. And here is a partial list of some of the almost obvious gains that you might have if you're able to build a full interactive computerized model of an animal, uncover gaps, correct errors, form theories, of course predict new phenomena and suggest experiments to be done in the lab, discover emergent properties, which are things that kind of emerge out of the model on their own, verify theories against observations, and the sky's the limit, synthesis, drug construction, and so on. How to do the modeling? The brief answer is I don't have time, of course, to give you the techniques and to show you what languages and tools we use from computer science and software engineering to do this. But one thing I do want to say and show you in a minute is make it look good. We don't want numbers and colored dots running across the screen. And I'm going to give you now some very modest examples of scratching the surface of this challenge, not in the sense of trying to do a whole animal, but in the sense of trying to capture some complicated phenomena. The first project is something to do with the thymus gland, and where we modeled several thousands of T cells behaving in the thymus gland. An enormous amount of knowledge, not data, was somehow put into this project, which took almost 5 years to do. So you'll see several thousand cells. Each of these cells has a very, very complicated program So it's an enormously complicated computation going on here. In a minute, on the left, about a third of the screen on the left, you can start seeing cells kind of conjugating around these long lines, which are epithelial cells. The details are not important. But the thing that's going on the left third of the screen is an emergent property whereby these cells kind of elbowing themselves in order to try to carry out some chemical activity that causes the lucky ones to reach the right-hand side of the screen, almost the right-hand side, and become fully fledged T cells. The black X's, the black crosses are apoptosis. You see cells dying. And again, the details are not important. And now if we go to the next slide, I want to show you that the previous one was really just a run of the system that we recorded. But you can actually interact with the system if we can run this one now. So this is just an example of how we sit in front of the screen and we can make changes in the execution itself. So we can zoom in further, we can see exactly which cell is the parent of any other cell. If you look closely, you'll see some of the receptors protruding, those are active. Now we right-click one of these cells and we make changes, so you're actually treading on the elephant's foot, so to speak, changing in this case the receptor status. This is all done. This is just a recording of how we played with the system. And now you see suddenly one of the receptors growing. We just made that change. And now we can go on to the next one. So that was an example of how you don't run this yet. I just want to say a few words. This example is the same system run under two different sets of initial circumstances. The first one is a wild type run. And in the second one, we put in a change, I don't want to get into the details, which we had a feeling was very important in deciding that these cells really compete to become T cells, and they have this emergent property on the left. Let's see this now in operation. On the top screen, you'll see a run very similar to the one we just saw. It's just recorded a bit more slowly. On the bottom is the same system where we switched off a couple of things that we thought were relevant to this. And what you'll see on the top in a moment, again on the left third of the picture, you will see again these hundreds and hundreds of cells, some of them committing suicide to let their siblings move ahead, and the others competing fiercely to carry out these interactions with the epithelial cells. On the bottom, almost nothing like that is happening. And I like to say it's a bit like if you take us scientists and you switch off our scientific competition genes. You know, I probably wouldn't have traveled to Berlin today. Maybe these gentlemen would have been playing golf or sitting with their grandchildren and playing. The fierce competition that we have in order to break down the walls and do our science better comes from something. And here we made the change in the biology, and we got a very weak development of the thymus gland. Of course, the mouse, happens to be a mouse, its entire immune system would collapse if this were really the case. But I'm showing you this in order to give you an example of how you can do experimental biology in silicon and actually see on the screen the difference in the first experiment versus the second. Another project I want to show you a video of is the pancreas. Here we did organogenesis, which is trying to capture how an organ grows. As a side remark, which is not on the slide, this year is the 100th birth of Alan Turing. A lot of you know Turing from Second World War. He also did a lot of work on biology. So if we can run this, I heard I'll just show you the beginning of this. This is a pancreas growing, and again the programming was done for each cell. The cell has a program. You throw several thousands of these cells together and you get the shape of a pancreas growing essentially out of nothing. I'm going to skip this forward so that I can spend my last minute concluding, but this kind of goes on and becomes more elaborate. You can see on the bottom what the model looks like versus what the real thing looks like. You can see the similarity. And we also had some, I think, rather amazing insights into why a pancreas looks like a pancreas, and a lung looks like a lung, and a liver looks like a liver. And we were able, in the same model, to make changes to the computerized model and to get the same system to look like a liver on the left or like a lung on the right. And the idea that I want to put forward is to build a complete model of the roughly 1,000 cell C. elegans nematode worm, which, as you can see from the video on the right, is completely transparent. And as you can see on the left, We actually know exactly how it develops, at least in video format. There's an enormous amount of information about this 1,000-cell animal, and this is something I would like you to be able to give water to a speaker in 10 years' time at the Falling Walls conference and show an animated video of the C. elegans nematode being completely modeled, so that what you see here is not going to be a worm, but is going to be a fully computerized model from which we will understand a lot about life. Cheers.